Hi, I'm Alan. Thanks for the intro, Jim. Today we're going to be talking about here's the agenda. This is, I think, less of a mixed audience than the last one I presented this for, so we can skip over some of these early slides about what's this motivation behind big data, especially for the technical nature of some of these other talks. Also, these particular solutions in the ecosystem, I'll just touch on some high-level MapR stuff. NoSQL and Spark, I think, are mostly known in the room. And then I'll get into some specific biotech use cases, which I think will be the interesting part for you guys, specifically around some population databases and data warehouse work. And then I'll talk about porting legacy workflows. This is a common problem we see in high-performance computing. How to take some existing legacy components and bring them into the new paradigm for better execution. So this idea of data inertia, this is really the fundamental thing behind why we're building these distributed systems today, and why there's this re-architecture that's happening of sending the compute over to the data. And it's basically because the network is the limiting component of a system when you can't keep all of your data on a single node inside of a single compute context, and it's slow to move the data between nodes because the network is relatively slow compared to the buses and the caches that are inside a single chassis. So the traditional way of doing computing is more of a naive approach. This is usually what's taught in standard algorithms, the factoring, where you have an algorithm that's executing in some loops, let's say, procedural program, and you're pulling data into the algorithm from wherever the data that needs to be operated on is stored. And so this is very much in alignment with the traditional way of distributing computing jobs on a high-performance computing cluster, or an HPC, I'll call it sometimes, where you have an HPC cluster on one side, and you have a SAN on another side, and you have a high-speed interconnect between them, and you read data in from the SAN into the computing cluster, you do your work, and then you write the data back out. And so that connection between the two of those becomes critical, because you can only move as quickly as you can move data between the two. There's an assumption I'm making here that we're talking about IO intense computing here. I'm specifically not talking about CPU intense computing. So the key insight in this kind of IO intense computing problem is that as the amount of data increases, this is going with Moore's law, or even exceeding it in the area of imaging, sensors, and DNA sequencing, the relative cost of moving the program to the data becomes proportionally less than the cost of moving the data to the program. So it becomes increasingly attractive to refactor things into this kind of big data style. The modern style. So there's a couple of technologies I'll be referencing here. One of them is Spark. People are generally aware. Can I just see a quick show of hands? Some people in the room? Great, okay. So it's an in-memory computing framework, runs on a cluster, scales out. You write the program once, it can basically scale out to thousands of nodes, as we've seen at MapR with our customers. HBase, people are familiar with NoSQL, I assume, since that MTEL was just giving a talk about that. It's a way that you can, well, I won't get into it. And so, MapR is combining these technologies onto a single platform to improve operational characteristics, making it more stable, make high performance, etc. So now I'm going to get into a use case coming from one of our customers for using this kind of system. This is specifically a batch data loading use case, and the problem here is a very common one. This doesn't just show up in pharmaceuticals or life sciences specifically. It's a problem across data warehousing generally. So this is a horizontal use case across many industries. So I thought this would be useful to not keep things too life science specific for the audience. The problem here was they were doing a data ingest as part of the R&D pipeline that's generating insights, generating candidates for feeding into their candidate pipeline for developing new therapies, new drugs. And it's all kinds of data, video data, proteomic data, next generation sequencing, DNA, RNA sequencing data, metagenomic data, you name it, it's all in there, electronic health records. And what they're finding is that their data warehouse was at maximum capacity. They'd already increased the size of this thing, but and tried to improve the procedures that were running inside of the data warehouse environment to do the data load, and that optimization just wasn't helping anymore, and the data were still growing. So this led to a problem for the execs that were taking care of this. This is CIO's office that was decreasing or not allowing the rate of insights coming from the data to be sustained or increase, which in turn led to a career limiting bottleneck. So this is not a good thing. 3 options to solve this. You can basically say, okay, well, we're just not going to have any more insights or candidates coming out of this pipeline. Obviously, that's not going to be popular with people. You can increase the EDW size. Since again, we know the procedures were already optimized to squeeze the performance out of it as best they could. But this is expensive because, again, the data are accelerating in terms of how quickly they're growing and outstripping the rate at which the hardware costs are coming down. And this is also not known to scale particularly well because the EDW system was not designed for good utilization in terms of a scale-out model, like we're talking with Spark or a NoSQL solution. So the third alternative was to find a more scalable solution, and we'll talk about what that looked like now. So here's what the original data flow looked like. There was some raw data coming in. I'm showing here raw data, public-private records, compound data, EHR data, et cetera, going through an initial cleansing step and getting loaded into a data warehouse. And then from there, the data were again transformed and loaded to some downstream, to a downstream knowledge graph database. And then that was being used to project out to data marts and other reporting systems, and also being used for downstream research applications. So it's really this, I guess I don't have a laser, but it's that first cylinder. Oh, there's a dashed line around the data warehousing environment. So this is the thing that's causing the problem. And some further analysis here shows that actually the majority of the space, again, that's the billing model for an EDW is based on terabytes consumed. The majority of that space was actually being used for operational purposes, in that it was temporary tables being used for joining the data. These data are particularly difficult to join because frequently you don't have a good identity that can be used to match the records. So there's a lot of auxiliary fields and a lot of auxiliary matching you need to do in order to figure out roughly what records correspond to what other ones, and it just kind of explodes the problem, especially as you have, again, each data set is larger, and then there's a plethora of data sets, so it's highly dimensional. And getting, again, increasing the capacity didn't yield an improvement, primarily because that join that was being done across this, very highly dimensional, large dimension problem was not designed to scale out across the system. So the alternative, and this is actually what was selected as the solution to the problem, was rather than sending the raw data into the data warehouse environment directly, was to send it into a staging area. This solution, if you want to look up case studies online, is called an ETL offload. And the reason is we're taking this initial extract and load step, moving it onto a scale-out, low-cost commodity cluster running a Hadoop system, in particular a MapR one in this case, and doing that extraction and operational EDW part of the process in the Hadoop space. And then once the data are clean and joined and reduced down, only moving that into the data warehouse environment. And then for further transformations on those data, like augmentation of things that need to go into the knowledge graph, that was also moved over into the MapR system. So now we have 2 dashed lines. There's this MapR, pre and post EDW phase, and then the data warehouse really is just going back to becoming a system of record. It's no longer responsible for doing any online analysis, either ahead or either on the operational side for loading the database, or downstream for driving into the knowledge graph and feeding this research pipeline. Was there a question over here? Thought I saw a hand. I guess it's just cameras. Sure, take pictures. I'll share the slides though too, so don't feel like you need to capture everything. There will be a link at the end. So the simplified analysis of this is that, again, it's a lower cost per terabyte to use this commodity hardware. to increase the operational capacity that you have, and so that's also more appropriate for the workload as well, because the reason that the EDW again is not scaling out is because it's not doing a join in a distributed manner, whereas specifically that's what Hadoop and Spark are particularly good at, is doing distributed sorts and distributed joins. Right, it scales out, it's basically just linear as you add more nodes. And it also introduces a strategic advantage, because having this kind of platform in place doesn't just solve EDW problems, it also solves problems that allow you to even bypass the warehouse entirely. So you might imagine there's some other interesting things you could do with these data that are coming in a more raw form before they're cleansed, such as like with the EHR data, where it's very unstructured and it's maybe not even entirely clear how you would normalize it into the data warehouse. You can just start processing it here in the Hadoop side and fast lane your data directly up to the R&D pipeline for self-service, as opposed to trying to shoehorn everything into a relational schema. So this is another benefit that came out of it. All right. Second use case I'm going to talk to you about, this is a scalable population database. So what's happening, just to give you a bit of background, cost of genome sequencing is coming down very, very quickly, faster than semiconductor improvements. And so they call this even Moore's law sometimes. It's something like a 400% compound annual growth rate. And so what you'll see if you watch the media in this space is that the number of people being included in these public studies is just going up really quickly. Just in the last couple of years, it's gone from 10,000 person studies to over 1 million person studies are being announced now. And eventually, everybody's going to be sequenced. It'll be end game once you see that they start announcing that newborns, when they prick the bottom of their foot to draw the blood to test for allergies and sensitivities and metabolic syndromes and so on, they're just going to do the DNA sequencing. That's game over. That means everybody's sequenced. And so it'll just take a couple of generations, and then we'll have full pedigree of all humans. This is not the only area that's doing this, though. It's particularly interesting for agricultural industry as well, precision agriculture, because while for humans mating is effectively random, and we can't do directed domestication, we have no problems really worldwide of doing directed breeding and domestication for livestock and plants. So there's what these companies are effectively trying to do is find some prognostic or for humans as well, find prognostic or diagnostic biomarkers for in healthcare circumstance it would be for pharmacogenomics. So this means you want to be able to identify the causal relationship between a genetic variation that somebody's carrying and what that will how that will present itself in a clinical context for their response to a drug, their propensity for developing a disease, and so on. So you can have a good prediction of what's likely to happen and what's the most efficacious way to intervene in an adverse scenario. So the idea is, again, to basically find a bunch of these genetic variants, whether they exist in the wild population like with humans, or they exist in a domesticated population with plants, or in an engineered population of synthetic organisms like some people are doing around here, and to understand really what does that carry through to on the phenotype side in terms of what's the yield or the benefit or the downside of having that particular variant. And this is fortunately a well-studied problem from another domain, and I'll get into that. Essentially what this means is that if you have a population that you're trying to categorize like this, you can think about the dimensions of the problem really being some deltas, which I'm showing on this y-axis here, and then some phenotypes, which are the way that the organism will present based on that genetic variation. And the phenotypes are on the x-axis. And then you're looking for co-occurrences between the two. So this is kind of a simplification, but this is generally how you would think about such a thing. And the more, the size of this ball I'm showing here at the intersection means that there's a higher co-occurrence of those two particular things in that two-dimensional space that I'm showing.
What is a sparse billion? Sparse?
Oh, because, well, there's potentially some uncountable number of phenotypes. This is very highly complex, and we don't even really have a good grasp on it for humans, let's say, but for a plant, you could look at, let's say, its rate of growth per day, or its number of grams of carbohydrate yield per head, or something like that. And then on the other dimension, sparse billion numbers of genotypes. This is basically if we were to iterate over all possible mutations along, let's say, human, which has about 3 billion positions in the genome. That's assuming you only do single base changes. You're now getting into 4 to the 3 billionth possible combinations of genotypes. So it's a very huge exploration space, but fortunately, because of some pedigree-related effects I don't want to get into that's fundamental to the biology, we don't really need to explore the whole space. We can reduce this down to just something much smaller, but it is still quite large. And what you can do when you encode the data like this is you can use a standard linear algebra technique for creating a low-rank approximation of the matrix. And one of the key operations you'll do there is to create these things called eigenvectors, which are basically just ways to describe what are the general patterns that we see in this matrix. I'm sorry I don't have something more illustrative about this to describe it, but let's focus on the business context. And so really what this is doing is it's giving you, for a row eigenvector, it's basically giving you a set of related phenotypes that are related to a particular SNP, and then a column is the transpose of that. So it's a set of SNPs that are related to a phenotype. And fortunately, there exist libraries in Spark and as well as Hadoop using a library called Mahout that allow you to do this kind of thing. MLlib is the one that you would use in Spark, and that's the more common one these days. If you're familiar with the concept of a GWAS, this is a genome-wide association study. So for those of you who are more in the clinical or life sciences side in the room, this is basically just a description of how a GWAS works. We're just finding a bunch of genes that are associated with a phenotype, and the transpose is a PheWAS. So this shows you how there's a bunch of phenotypes associated with a particular genetic variant. I also want to point out that doing this type of operation is a mixed I/O workload, so it requires both high throughput sustained reads as well as random access. So you need it's a mixed workload, and this is particularly suitable for MapRDB. There's another use case that I don't have time to get into today, but that's pretty interesting, which is the largest biometric database in the world that's been built to date. It has about 1 billion people in there. This is a national identity program in India. So what they're doing is they're scanning all the irises of everyone and scanning their fingerprints, and they're building a database that allows authentication at, say, an ATM or going to an aid disbursement program for fuel or for food to identify. The reason to build this is because there's been a lot of fraud in that system, and it's leaking a lot of money where the resources aren't actually getting to where they need to go, or they're getting caught by some corruption in the system. And so the interesting thing here is that as you're adding more people into the database, you need to constantly be doing this something like a matrix factorization, because every time you put more people in, it changes the meaning of what's already in there. If you find somebody who has two people who have very similar fingerprints, what you were previously using for the first person to identify them may no longer become the same features that are valid because you have two fingerprints that are very similar to one another. Anyway, interesting use case. Happy to talk to anybody about it at the booth if you want to get deeper into that. Anyway, okay, so what I was going to say though is that if we just change these labels to users and documents, then the eigenvectors that fall out of this are interests and behaviors, so user behaviors or user affinities for particular types of content. And this is effectively how Netflix works for recommending movies to you, or how the big three on the internet are generating their revenue, because they need to figure out how to contextually show you the correct offers so that you click them and put money in their coffers. So when I said this is a solved problem, I was saying that it's a solved problem in terms of once you abstract everything away from the business jargon specifics and map it into the math space and start to look at what do the data access patterns look like and what kind of computing problem you would use to solve such a problem, it's really the same problem. All right, so we can do this, we just need the data. Okay. So the last thing I wanted to talk about, hopefully I can get us back to close to on schedule, is porting some legacy pipelines. And there's actually a full-blown like 30 or 40-minute presentation on this. I'm just going to give you the highlights, and that's available as a recorded webinar and slides on my SlideShare, which I'll show you later. And also a GitHub repo, which I'll link to also. So the idea here is, are people generally aware, like, I didn't do a poll of life sciences people in the room. People are aware, like, what genome sequence alignment is, generally? Okay, very few. So there's a problem when you want to do some DNA sequencing. It doesn't, and I mentioned in response to your question, someone over here's question earlier about why was I talking about billions of yeah, that huge sparse matrix, thank you. When you're reading a genome, you're not really reading out a long string of 3 billion, 3 gigabytes long. It's really coming out as lots of little fragments. And so these fragments need to be reassembled to map back onto what's called a reference sequence to figure out where this person that was currently sequenced differs from something that's considered as a reference. So it's all relative. And these genetic variants are relative to a reference. So there's this operation that needs to take place called an alignment, where you're taking some small, I'm trying to use my laser pointer. Okay, I have to stay here. Taking some small reads, they're usually on the order of a few hundred base pairs long, and then aligning it to some reference sequences, which are usually on the order for humans of hundreds of millions of base pairs long, and getting some aligned reads out of that, and then those aligned reads again go to some downstream stuff we don't care about for the sake of this discussion. This particular piece, let's focus in on here. There's 3 ways that it can come out. For something that you don't know whether it aligns, it can either align to a single location. It's like, okay, great, we know this read came from the human genome, and it's unambiguous where it came from according to the reference. You could have some ambiguity in terms of the alignment. Maybe the read is too short. Maybe it aligns to an area where there's many sequences that look very similar to it, given the error rate that we know that's associated with the DNA read. And then there's other cases where things are just unlocatable. So that could be a situation where there was some contamination, for example, where there got to be some, maybe somebody was eating some corn chips in the laboratory and some corn got into the tube, and now you're trying to align the corn to the human genome and it's just not working out. This is actually like realistic. Happens all the time. Corn DNA is everywhere. So what you have is a many-to-many relationship. A read can be at multiple locations. A read can exist in no location, and a location can have multiple reads. In terms of what this looks like for parallelization, so this is traditionally how things have been done, and is actually how it's being done in most places that are doing these kind of pipelines today. You have this big file of DNA reads. You either partition that file, or you pass something into your scheduler for the HPC cluster that tells it how to partition up the file. On the left, so we're going to do a split. And then there's an align operation that happens that's trivially, like embarrassingly parallelizable. You can just basically farm it out across the HPC cluster. It's going to generate 0 or more locations per read, per partition. And then what happens is that's going to create a local output on your HPC cluster and local I/O. But then what happens is all those data need to be pushed back to a central storage. So you can see now all these arrows are kind of converging down to a single point. That's going to create a hotspot, and that's that bottleneck that exists I was talking about before between the SAN cluster and the compute cluster, and that's going to take a lot of time. The other thing that needs to happen is that sort operation. For an HPC environment, there's not really a built-in primitive there for doing a distributed sort. So that sorting all needs to happen on a single node, which is going to also bottleneck things, because you have a single node doing the work that was previously, parallelized across a number of machines, and that can be very difficult to get a good level of utilization across your cluster if you don't have many use cases running at the same time in like a multi-tenant environment, because this one pipeline is going to be blocked just by that single step. What this is going to put out is a list of ordered aligned reads. That's again useful for the downstream application. Don't worry about that. And whatever happens after, again, we don't care. I should have just shown you this slide a moment ago. Right, so there's a volume read bottleneck here. When we have the SAN and we're trying to suck all the data onto the HPC cluster, it's not really too bad. But this write operation and this sorting operation is the one that really just kills the system, where the cluster will be mostly idle with a single node very hot for a long time. At a high level, this is basically what Spark or Hadoop is allowing it to do, is it eliminates this split step up front, because the data are just already internally distributed across a number of nodes. And it's not necessarily something a user or an application programmer needs to worry about. They're just kind of pointing at some data somewhere. And it doesn't really matter to that user, how the data are organized. They just need to know that they have some kind of handle to it and they can operate on it. The alignment, as with the HPC example, can take place in parallel. That's all very easy. But then the nice thing is this, again, because these big data style systems can do a distributed concatenation and sort, you don't need to bottleneck down to a single node to get to your final result in the scope of the problem we're talking about here. It can just be done also in a parallel manner. So I told you I was going to talk to you about some legacy stuff. The nice thing is, so there's these legacy processes that are sort of assuming that the data will come to them, and they are not, as a program, going to be distributed to the data. It's kind of an inside-out factorization of the problem. And there's a lot of legacy code that exists from academia in a high-performance computing environment. This is not just a problem with biology or genomics. It also exists in physics and combinatorial chemistry, and pretty much anywhere where the academia is very close to industry and there's just like a little missing applied science place. So anywhere where a lot of things in San Diego are related to this, actually. You have these kind of problems of unmaintained code from students that just wanted to finish their dissertation, graduate, and get a professorship. And it's difficult to deal with when you want to operationalize it. So fortunately, what you can do is you can, in Spark, they have this, the data structure that's being used to pass the data around between these steps. So the kind of in-flight thing in the yellow arrows is called an RDD, redundant distributed data set, I think. Resilient, thank you. I lose track of the acronyms. I confuse that one with RRD, which is a round-robin database also all the time. Yeah, so that's kind of this yellow piece here. And what it has is an operation on it called a pipe, which allows it to basically call it a subshell. So this is a way that you can create a little local computing context, which I've drawn a gray box, dashed line box around here. And then you would launch that legacy application inside that sandbox to do local processing. So there it thinks, it doesn't understand that it's been captured and put inside of a box. It's just going to do its normal thing, and it's going to do it in a local I/O context. When it finishes up, you can have your process that set up that sandbox tear it down, pull the data back into an RDD, and then send it back into the Spark workflow. And the generalization of this is, of course, if you have a long, complex workflow, you build a bunch of these things. Some of your steps are calling down into these gray boxes, as shown on the left and the right columns, and in the middle, we're just doing like a native Spark thing. So ultimately, what you'll find is that, kind of similar to where we talked about this EDW problem, there's going to be some hotspots in the code. You don't necessarily want to convert everything to Spark, like this step in the middle I'm showing, but to profile the code, figure out which of these gray boxes are particularly problematic, and then just port those, and over time bring things into a new paradigm. And so this kind of design pattern buys you the flexibility to do that, to focus in just specifically on the pieces that are causing problems for your operational system. Again, I want to keep us on schedule as best I can. I don't have time to get into the code for this today, but there is a slideshow specifically about this. This slide deck, as well as many others on this and similar topics, are at slideshare.net/Alan Day. And particularly, there's a code base that you can grab, and it's got the README basically is copy paste. So if you have Spark running locally and you have some, you have Git, you can just run it and it will download some data and process it using a liner called Bowtie and show you how that all integrates with Spark. It includes the glue code for connecting for these little connector pieces. This is kind of the non-obvious tricky part, integration logic that I demonstrate. And that's the end of my show. So you can follow me on Twitter, you can email me, LinkedIn to me, whatever you like. Thanks. No time for questions. You can find me in the back. Thanks.
All right. Thanks a lot, Alan. During lunch, we'll also be networking, time to talk.